All right, everyone, welcome to another deep dive. Today, we're tackling Coxiella burnetii.
The bacterium behind Q fever, right?
Exactly. And our source material is Gillespie and Hockey's Principles and Practice of Clinical Bacteriology. Quite the weighty tome. But no worries, we'll break it down. This deep dive is especially for all you medical microbiologists out there.
Excellent. It's always a pleasure to delve into these fascinating microbes.
But Coxiella burnetii, what's the first thing that pops into your mind when you hear that name?
Well, it's a bit sneaky, isn't it? Coxiella burnetii is a tiny gram-negative bacterium, and get this, it's an obligate intracellular bacterium.
Meaning?
It can't survive on its own. It has to live inside a host cell to replicate, like a little cellular freeloader.
So not just hanging out on surfaces waiting to pounce.
Nope, needs a host cell to hijack. And the thing is, it's not picky about what kind of cell it infects. A wide range of cells in both humans and animals.
Okay, that's a little unnerving. And claim to fame, so to speak.
Q fever, a zoonotic disease, so it can jump between humans and animals. And clinically speaking, it's a chameleon. It can present in so many different ways.
A diagnostic challenge, then.
You could say that. Keeps you on your toes, for sure.
So, under the microscope, what does this stealthy bacterium actually look like?
Well, Coxiella burnetii is small, rod-shaped, typically less than 1 micrometer long. But honestly, it's difficult to see with your usual staining because, it hides inside cells.
The stealth operator we talked about. No wonder diagnosis can be tricky. And how about its mode of infection? How does it actually get into the host?
Inhalation, primarily. Contaminated aerosols. Think like dust or droplets containing Coxiella burnetii shed from infected animals.
No.
Mostly urine, feces, birth products.
So not just direct contact with animals, it can be airborne?
Exactly. And here's the scary part. Only a tiny amount is needed to cause infection. Studies suggest inhaling as few as 1 to 10 bacteria can be enough to make you sick.
Wow, that's a very low infectious dose.
Yeah.
Once these tiny bacteria are in, where do they head? How do they actually cause disease?
Oh, macrophages, those immune cells that usually protect us by engulfing invaders. But Coxiella burnetii is clever. It tricks the macrophages into taking them in.
Like a wolf in sheep's clothing, using our defenses against us.
Exactly. It survives and replicates right there, inside the macrophage, safe and sound.
So it's not just hiding, it's actively setting up shop.
Oh, absolutely. Inside the macrophage, it transforms into a different form, the large cell variant. This form is metabolically active and starts replicating like crazy.
So it's like it's got two distinct forms, the small cell variant and the large cell variant. What's the deal with that?
It's all about strategy. The small cell variant is the tough guy, the infectious form. It can survive outside a host for ages, heat, drying, even some disinfectants don't face it.
A tiny armored vehicle built to deliver that infectious payload.
And the large cell variant, all about replication. Once inside a host cell, the small cell variant transforms and starts churning out copies of itself, like a little bacteria factory.
So, a remarkable adaptation to ensure survival and spread. But let's bring it back to the patient. What are the signs someone might have Q fever?
That's where it gets tricky. Q fever can present in so many ways. Some people are asymptomatic and don't even know they're infected.
Silent but deadly.
Well, not always deadly, but you get the point. Then there's acute Q fever, which often looks like the flu fever, chills, headache, fatigue, muscle aches, you name it.
So easy to miss or dismiss.
Exactly. Especially if they don't know they've been exposed to Coxiella burnetii. People might think it's just a bad cold or the flu.
Patient history is critical then, right? Asking about contact with livestock or animals.
Absolutely. Knowing the potential risk factors helps a lot, especially in guiding testing.
So let's say we suspect Q fever. What tests are available to confirm?
Serology is the gold standard, looking for antibodies against Coxiella burnetii in the blood. Basically, checking if the body's immune system has raised the alarm.
Makes sense. What kind of serological tests we're talking about?
The indirect fluorescent antibody, IFA test, is the most sensitive and specific. It involves fixing Coxiella burnetii antigens to a slide, adding the patient's serum. If antibodies are present, they bind to the antigens, and we can see this binding under a fluorescent microscope.
So, a visual test, looking for that fluorescent signal. Any other options?
There's the complement fixation test, a bit more complex, but it's based on the principle that when antibodies bind to antigens, they activate a cascade of proteins in the blood, the complement system.
So we're not directly visualizing the antibodies, but observing the consequences of their presence.
Exactly. And a key point to remember, both IFA and complement fixation use different phases of Coxiella burnetii antigens. Phase I antigens are from bacteria isolated from animals, while phase II antigens are from bacteria grown in cell culture.
Why does the phase matter?
Because the antibody response can vary depending on the stage of infection, acute or chronic Q fever.
Okay, so the phase of the antigen helps with interpretation. Serology sounds nuanced. What if it's inconclusive or we need a faster result? You mentioned PCR earlier.
Yes. PCR, or polymerase chain reaction, can directly detect Coxiella burnetii DNA in clinical samples like blood or tissue. It's incredibly sensitive and can often provide a diagnosis much earlier than serology.
So we're bypassing the antibody response and searching for the bacteria's genetic fingerprint.
Precisely. It's especially useful in chronic Q fever, where the antibody response might be weaker, or when we suspect infection in tissues that are difficult to biopsy.
So PCR is a valuable tool in the diagnostic arsenal. What about actually culturing Coxiella burnetii? Is that done routinely?
Not really. It requires specialized facilities and expertise. It's an obligate intracellular pathogen, remember? It needs to grow inside cells. Plus, there's a safety concern because Coxiella burnetii can be transmitted through aerosols, posing a risk to lab personnel.
So it's more of a research tool, though.
Yes, primarily used for research, and even then, it's challenging. Often involves inoculating cell cultures or embryonated eggs with patient samples and then monitoring for signs of growth.
Sounds time-consuming and delicate. So culturing is important for studying Coxiella burnetii, but not for everyday diagnosis.
That's about right.
And speaking of managing the infection, our source mentions doxycycline as the primary treatment for acute Q fever. Why is that the go-to choice?
Well, doxycycline is a tetracycline antibiotic,
Yeah.
and tetracyclines are quite effective against Coxiella burnetii. They work by inhibiting protein synthesis in the bacteria, basically stopping them from growing and multiplying.
And is doxycycline usually well tolerated?
Generally, yes, and it's readily available, making it a practical first-line treatment.
So effective, safe, and easy to access. But what about chronic Q fever? Are tetracyclines enough for those cases?
Unfortunately, no. Chronic Q fever is a different beast. The bacteria can become entrenched in tissues like heart valves, bones, even the liver, and they can enter a dormant state, making them less susceptible to antibiotics.
So they're hunkering down, building a fortress, making it harder for the antibiotics to reach them.
A good analogy. And chronic Q fever often requires a combination of antibiotics. Usually, doxycycline is paired with another drug like ciprofloxacin, a fluoroquinolone.
So a two-pronged attack. And how long do people typically need these antibiotics?
That's the challenging part. Treatment for chronic Q fever can be very prolonged, lasting for months, even years. The duration depends on the severity of the infection, which organs are involved, and how well the patient responds.
So it's a marathon, not a sprint, requiring patience and vigilance.
Absolutely. It really highlights the importance of early diagnosis and prompt treatment for acute Q fever to prevent it from becoming chronic.
To stop those bacteria from getting a foothold, let's shift gears to prevention. What strategies are there, both on an individual level and from a public health perspective?
Breaking the chain of transmission is key. For those at high risk, like people working with livestock or in labs handling Coxiella burnetii, personal protective equipment is crucial. Masks, gloves, gowns, anything to prevent exposure to aerosols and contaminated materials. And vaccination. Highly effective and recommended for high-risk groups.
So, shielding yourself from those sneaky bacteria. What about broader measures?
Public health surveillance is vital. Tracking cases in both animals and humans can help identify outbreaks early and implement control measures quickly. Education is also key, especially for high-risk groups. They need to know about transmission routes, preventive measures, and the importance of vaccination.
So knowledge is power. And it sounds like collaboration between healthcare professionals, veterinarians, and public health officials is essential to manage this zoonotic disease.
Couldn't agree more. It's a team effort requiring vigilance, communication, and a comprehensive approach to keep C. burnetii in check.
And on that note, we'll wrap up this part of our deep dive into Coxiella burnetii. Stay tuned for part 2, where we'll continue exploring this fascinating and challenging bacterium.
Welcome back to our deep dive on Coxiella burnetii.
Glad to be back. Now, before we move on, I wanted to circle back. So it's not just about the duration of the infection. It's about where those bacteria decide to set up shop.
Exactly. Heart valves are a common target, but they can also infect bones, liver, even the brain in some cases.
Sounds pretty serious.
It can be, unfortunately. Once it sets up shop in those tissues, it can be really difficult to get rid of. Antibiotics might not be able to penetrate those sites as effectively, and the bacteria themselves can go into a kind of dormant state, making them less susceptible to treatment.
So, harder to reach, harder to kill.
You got it. And the consequences of chronic Q fever, well, they can be pretty severe. Endocarditis, an infection of the heart valves, is a particularly worrisome complication, can lead to heart failure and all sorts of other life-threatening conditions.
Wow, that's a scary thought. It really emphasizes the importance of early diagnosis and treatment, right, to prevent it from reaching that chronic stage.
Absolutely. The earlier we catch it, the better the chances of successful treatment with a shorter course of antibiotics.
So let's talk more about treatment options. We know that doxycycline is the first-line drug for acute Q fever. But what if, for some reason, doxycycline can't be used? What are the alternatives?
There are a few other antibiotics that can be used, although they might not be as effective as doxycycline. Macrolides like erythromycin or azithromycin are sometimes used, but they're generally considered less effective against C. burnetii.
So doxycycline is really the gold standard whenever possible. Now, what about those chronic cases? Is there any hope of a cure?
It's trickier, but yes, there's still hope. As we mentioned before, chronic Q fever often requires a combination of antibiotics, and doxycycline is usually part of that. It's often paired with another antibiotic like ciprofloxacin or rifampin.
Hitting it from multiple angles.
Exactly. And how long treatment lasts really depends on the individual case.
I imagine it can vary quite a bit.
It does. Chronic Q fever often needs prolonged treatment, sometimes for years, and even then, complete eradication isn't always guaranteed. Close monitoring is essential to see how well the treatment's working and to adjust as needed.
So it's a long and winding road, requiring a lot of patience and careful monitoring.
That's a good way to put it. It really underscores the importance of prevention.
Speaking of prevention, are there any vaccines available to protect against Q fever?
There is a vaccine, but it's not routinely recommended for the general public. It's mostly given to folks who are at high risk of exposure, like people who work with livestock or researchers who handle C. burnetii in the lab.
So it's a targeted vaccination approach.
Yeah.
Makes sense. What about those of us who aren't in those high-risk professions? Are there any steps we can take to minimize our risk?
Just being aware of the potential sources of infection and taking some basic precautions can go a long way. If you're going to be around livestock, especially during birthing season, it's a good idea to wear protective gear like masks and gloves. And of course, always practice good hygiene, wash your hands thoroughly after any contact with animals or their environment.
Simple but effective measures. It's a good reminder that knowledge and awareness are our first line of defense against many infectious diseases.
Precisely. And this brings us to another interesting aspect of C. burnetii, its potential as a bioweapon. It's actually classified as a Category B bioterrorism agent.
Whoa, that's pretty serious. What makes it a potential threat in that regard?
Well, there are several factors. First, it's highly infectious, remember? We talked about that incredibly low infectious dose. Just a handful of bacteria can make someone sick.
Right, a little goes a long way.
Second, it's very stable in the environment. It can survive for long periods outside of a host, which makes it easier to weaponize and spread.
So not easily destroyed.
Right. Third, it can spread through the air, through aerosols. So it could potentially infect a lot of people very quickly.
That's a scary thought.
And lastly, it can cause very debilitating, even fatal illness, especially in those chronic cases we discussed.
So high infectivity, environmental stability, aerosol transmission, and the potential for severe illness, a recipe for disaster, unfortunately.
Exactly. It really emphasizes the importance of being prepared for a wide range of threats, including those posed by biological agents.
It's a lot to take in, but fascinating nonetheless. And speaking of taking in information, I think it's time to put your knowledge to the test. We've talked a lot about C. burnetii, so let's see what's stuck. Are you ready for some exam-style questions?
Beautiful.
Right, question number 1. What are the two distinct morphological forms of C. burnetii, and how do they contribute to the bacterium's survival and its ability to cause disease?
Well, we've got the small cell variant, or SCV, and the large cell variant, or LCV. The SCV is the tough one, the infectious form. It can survive all sorts of harsh conditions, both outside of a host and inside. That resilience helps it spread and initiate infection. Once it's inside a host cell, the SCV transforms into the LCV, which is metabolically active and focuses on replication. It's this two-pronged approach with a hardy infectious form and a replicating form that makes this bacterium such a successful pathogen.
Excellent explanation. It's all about playing different roles at different stages of infection. All right, question number 2. A patient comes in with a sudden fever, headache, and pneumonia. What are the key diagnostic tests to confirm or rule out Q fever? And why is choosing the right test so important?
In a case like that, you'd want to do a few things. First, a chest x-ray to confirm the pneumonia. Then, to specifically look for Q fever, we'd turn to serological tests. The indirect fluorescent antibody, or IFA, tests and the complement fixation test are commonly used. These tests look for antibodies against C. burnetii in the patient's blood. Choosing the right test is crucial because the type of antibody response can vary depending on whether the infection is acute or chronic. For acute Q fever, the IFA test is generally preferred because it's more sensitive in the early stages.
So it's all about using the right tool for the job, and that decision can be influenced by the timing and the specific symptoms. Okay, question number 3. We've talked about how C. burnetii is primarily spread through aerosols. But are there other ways it can be transmitted? And who are the people most at risk of getting infected?
While inhaling contaminated aerosols is the most common route, C. burnetii can also be transmitted through direct contact with infected animal tissues and fluids. This can happen during things like slaughter or assisting with animal births. There have even been cases of transmission through contaminated milk. People who work closely with livestock farmers, veterinarians, abattoir workers, they're at the highest risk. But anyone who comes into contact with potentially infected animals or their products can be at risk.
So it's not just an agricultural issue, it's something everyone should be aware of.
Yeah.
Okay, question number 4. We've touched on how tricky it can be to treat chronic Q fever. Can you explain why that is? And what are the potential long-term consequences if it's not managed effectively?
Chronic Q fever is challenging because the bacteria can basically wall themselves off within tissues, forming what are called granulomas. These granulomas can shield the bacteria from antibiotics, making treatment more difficult. The bacteria can also shift into a slow-growing state, which makes them less susceptible to antibiotics. If chronic Q fever isn't effectively managed, it can lead to some very serious complications. Endocarditis, hepatitis, osteomyelitis, even neurological problems.
So those persistent bacteria can cause a lot of damage if they're not dealt with. Early detection and aggressive treatment are really key here. And finally, question number 5. We talked briefly about C. burnetii's potential as a bioweapon. What makes it such a concern from a public health standpoint, and what steps are being taken to try to mitigate that threat?
Well, as we've discussed, C. burnetii is highly infectious, it's stable in the environment, and it can be spread through the air. This combination of factors makes it a very real threat if it were to be intentionally released. Public health agencies are actively working on this, though. They're developing surveillance systems to try to catch outbreaks early, stockpiling antibiotics for treatment, and researching new vaccines and therapies.
So a very proactive approach, which is definitely needed when you're dealing with a threat of this magnitude. Well, that wraps up our exam session for today. I hope you found it helpful. We've covered a lot of ground, from the basic biology of C. burnetii to the complexities of diagnosis, treatment, and prevention.
It has been quite the deep dive, and it really highlights how important it is to stay informed about this versatile and potentially very dangerous pathogen.
And we're back for the final part of our deep dives into Coxiella burnetii. We've talked about its stealth, its resilience, its ability to cause both acute and chronic infections. But what really fascinates me is its life cycle. It's like a microscopic thriller unfolding inside our cells.
It really is. Coxiella burnetii's life cycle is a testament to its adaptability and its ability to survive in really harsh environments.
So let's walk through it. Set the scene for us. How does this kind of drama begin?
Picture this. A small cell variant, an SCV, is just floating around. Maybe it's in dust, maybe it's in a tiny droplet of moisture. Someone inhales it, perhaps someone working with livestock, or maybe just someone walking by a contaminated area.
So at this point, the SCV isn't actively doing anything, just waiting for its moment.
Right. It's dormant, but don't underestimate it. These SCVs are incredibly tough. They can survive drying out, heat, even some disinfectants. They're designed to endure and eventually reach their target, macrophages.
Macrophages, those are the immune cells, right? The ones that gobble up invaders.
Exactly. They're like the body's security guards, and Coxiella burnetii has figured out how to exploit that.
So it's like a Trojan horse, sneaking past security and getting inside the fortress.
Perfect analogy. The macrophage sees this tiny SCV and thinks, oh, just a bit of debris, time to clean up. And it engulfs the SCV, taking it inside. But of course, that's exactly what Coxiella burnetii wants.
Because now it's inside the cell where it can do its damage.
Not quite yet. The macrophage packages the SCV into a compartment called a phagolysosome. Think of it like a cellular stomach filled with enzymes that are supposed to break down whatever's been engulfed.
So the SCV is in trouble now, right?
Yes.
Trapped in a digestive chamber.
You'd think so, but Coxiella burnetii has a trick up its sleeve. It actually thrives in this acidic environment.
It's like it enjoys being doused in stomach acid.
In a way, yes. This acidic environment actually triggers a transformation. The SCV, which was dormant, changes into the large cell variant, the LCV.
So it uses the macrophage's defenses to trigger its own development.
Exactly. And once it transforms into the LCV, it becomes metabolically active. It starts replicating, making more and more copies of itself within the phagolysosome.
So it's turning the tables, using the macrophage's own systems against it.
Precisely. Eventually, all those replicating LTVs cause the phagolysosome to burst. Now all those new bacteria are released into the surrounding tissues, and they can infect other cells, continuing the cycle.
It's like a prison break at the cellular level.
And to make things even more interesting, some of the LCVs actually transform again, forming spore-like structures that are even more resistant to environmental stresses. So they can survive outside of cells, too, waiting for their opportunity to infect a new host.
So it's a cycle of invasion, transformation, replication, release, and then the possibility of going dormant again. It's a pretty remarkable strategy for survival.
It really is. And it's this complexity, this ability to adapt and persist, that makes Coxiella burnetii such a challenging organism to study and to control.
Well, on that note, I think it's time to wrap up our deep dive into Coxiella burnetii. We've covered a lot of ground, from its basic biology to its complex life cycle, the diseases it causes, the challenges of diagnosis and treatment, and even its potential as a bioweapon.
It's been a fascinating journey, and I think it underscores the importance of ongoing research and surveillance to better understand and manage this truly remarkable and potentially dangerous pathogen.
Absolutely. We hope you've enjoyed this deep dive as much as we have. Stay curious, and we'll catch you next time on The Deep Dive.